Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Maharashtra State Eligibility Test for the subject Life Science. I will be discussing the question paper 2 part 2 paper, and it was conducted in the year 2021. So coming to the first question. Sodium dodecyl sulfate is used while separating proteins by polyacrylamide gel electrophoresis because it binds to protein and confer a uniform negative charge to the proteins, thereby making them move in the during electrophoresis. So the answer here is option B, that is it binds to proteins and confer a uniform negative charge density, thereby making them move during electrophoresis. Then next one, cytochalacin D inhibits the formation of microfilaments. Which of the following biological activities will not be hindered? So my muscle contraction, cytosolic transport of vesicles. Cytosolic transport of vesicle is through microtubules and not microfilaments. So option B, it will not be hindered. Amoeboid movement of phagocytic cell formation and cleavage furrow during following telophase of mitosis. Then next one. Which of the following statements for genetic drift is false? Genetic drift causes allele frequencies to change at random. Yes. Genetic drift can lead to richness of genetic variation within population. It doesn't lead to richness of genetic variation within population. So that is false regarding genetic drift. Then genetic drift can cause harmful alleles to become fixed. Yes. And genetic drift alleles affect alleles frequencies stronger in small populations. That is also correct. Then next one. According to Oparin, which of the following was not present in the primitive atmosphere of the earth? It is oxygen. Answer is option D. Life is said to have originated as coacervates that were formed by polymerization and aggregation. Answer is option C. Then when a taxon is restricted to a particular geographical region, then it is called. So it is located to a particular geographic region, so it is called as an endemic. Answer is option A. Severe combined immunodeficiency SCID mice do not have the B and the T cells. Answer is option A. The lichen rochella is a source of. So it is a source of dye that is royal purple color. Answer is option B. Then seasonal activity of cambium leads to. So it leads to both ring porous and diffuse porous wood. So ring porous trees, vessels laid down at the beginning of the growing season are much larger than those laid down at the end of the season. Whereas in diffuse porous woods, all the vessels are roughly the same diameter throughout the growing season. So seasonal activity of cambium leads to both ring porous and diffuse porous woods. The next one, cytoplasmic male sterility occurs as a result of interaction of. So cytoplasmic male sterility is total or partial male sterility in plants as a result of specific nuclear and mitochondrial interaction. So answer is option D, that is nucleus and mitochondria. Then next one, the enzyme acetyl-CoA carboxylase belong to which class of enzyme? So it belongs to the ligases. Answer is option D. Then according to Vavilov, mango, noble canes, rice and brinjal are some of the plants that originated in Hindustan center. Answer is option A. Then which of the following technique is best for determining the precise location of the radioactive isotope located in a specimen? It is autoradiography. Answer is option C. They have specially told location of radioactive isotope located in a specimen, so it is autoradiography. Then bacterial glucose phosphotransferase system. Glucose is converted to glucose 6-phosphate. What is the source of phosphate? So phosphotransferase system or PEP group translocation or PTS. It is a distinct method used by bacteria for sugar uptake where the source of energy for the system is phosphoenol pyruvate. So the answer is option C, that is phosphoenol pyruvate. Then next, The end product of adenosine monophosphate and guanosine monophosphate catabolism in normal humans is, so it is uric acid. The end product of purine metabolism is uric acid. Answer, adenine and guanine are purines. Then next one. Which of the following bonds in protein have partial double bond character? It is the C-N bond. That is option D. The peptide bond has partial double bond character and it is formed between C and N. Then when bacteria growing at 20 degrees Celsius and transferred to 40 degrees Celsius, they are most likely to synthesize membrane lipids with. So cells regulate their lipid composition to achieve constant membrane fluidity under various growth conditions. So bacteria synthesize more unsaturated fatty acid when cultured at low temperature. So when bacteria growing at 20 degrees Celsius are transferred to higher degrees Celsius, so they start at lower degrees Celsius they are synthesizing unsaturated fatty acid, means at higher degrees Celsius they will be having saturated fatty acid, that is of long chains. So the answer is option B. The type of microscopy we would use to examine bacteria that are stained by gram staining procedure is. So it is bright field microscopy, is also known as compound light microscopy. So the answer is bright field, normal lab microscope. So answer is bright field microscope. Then mesenteries are found in, they are found in triploblastic animals. Answer is option B. Then in a DNA sequence, if nucleotide A is replaced by nucleotide G, the resulting mutation will be, so a purine is replaced by a purine. So it is a transition mutation. Answer is option D. Transversion means the version would have changed. That is, purine would have become pyrimidine. So that is transversion. Then next one. Which of the following is correct for NAK ATPase? So here Na plus and ATP bind on the intracellular side while K plus and aubane bind to the extracellular side. That is option D. I have shown the picture in the next photo. Okay, this is the sodium potassium pump. Here in the first case the transporter binds 3 Na plus ions from inside the cell and ATP is getting converted to ADP. That is over here. ATP is getting, that is on the intracellular side. Then the transporter releases the 3 Na positive onto the outside and binds 2 K positive from the outside of the cell. Here 2 K positive ions are bound from the outside of the cell, and then there is a dephosphorylation and the transporter releases 2 K positive ions to the inside. So Na positive and ATP are bound from the intracellular side. So ATP and Na positive, they are bound from the intracellular side over here if you see the first picture. But aubane, now coming to aubane. Aubane binds preferentially to the form of the enzyme that opens to the extracellular side. So over here that opens to the extracellular side, this form is the one where aubane binds. So the answer is option D. Then next one. The sequencing of dash helped investigators conclude that all living things can be classified into 3 domains. The sequencing of cytochrome C. Answer is option B. Then mark the correct statement. In active transport, molecules move from higher to lower concentration. So during active transport, substances move against a concentration gradient from an area of lower concentration to an area of higher concentration. The next one. The carrier protein is involved in active transport and facilitated diffusion. That is correct. Carrier protein is involved in active transport and facilitated diffusion. Then energy is consumed to move molecules against a concentration gradient in active transport only. That is also correct. Then in active transport only water molecules are transported with consuming energy. In active transport actually substances, that is ions, glucose and amino acids, are transported with consuming energy. That is, they move across a membrane from a region of their lower concentration to a region of their higher concentration. Then next one, hybrid zone is a region where genetically distinct population meet and interbreed to some extent. Answer is option B. Then the sedimentation coefficient increases with. So sedimentation coefficient of a particle characterizes its sedimentation during centrifugation. So it decreases with partial specific volume. Answer is option D. So coming to the next question. Peptide antigen assembled with class 1 MHC aided by. So they are aided by chaperone molecules in the endoplasmic reticulum. This mechanism I have discussed in Maharashtra SET exam conducted in the year 2019. I have already made a video on that. You can refer that for explanation. I have given a picture also. Then next one. Type 2 glycogen storage disease, that is Pompe disease, is a genetic disorder in which the defective enzyme is. So it is an autosomal recessive metabolic disorder and it damages the muscles and the nerve cells. It is caused due to deficiency of lysosomal acid alpha glucosidase enzyme, which is responsible for the cleavage of alpha 1,4 and alpha 1,6 glycosidic bonds of glycogen to glucose. So the answer here is option C, that is alpha 1,4 glucosidase. Then next one. Which of the following is an example of an autoimmune disease? So it is myasthenia gravis, that is an example of autoimmune disease. Then, which of the following statements about leafy, a regulatory gene in Arabidopsis thaliana, is correct? So, leafy causes group of undifferentiated cells called meristem to develop into flowers instead of leaves. So, leafy is involved in floral meristem identity. So, the answer here is LFY is involved in floral meristem identity. The next one, binomial nomenclature include name of the plant in 2 words that designate. So, it is the generic name and the specific epithet. So, the answer is option C. The generic name, it identifies the genus to which the species belong, and the specific name or the specific epithet distinguishes the species within the genus. Next one. The causal organism for rice blast diseases. It is Magna porte grigia. Answer is option B. The center of origin of potato is Peru. Answer is option D. Helicobacter pylori uses urease to counteract a chemical defense in the human organ in which it lives. This chemical defense is, so Helicobacter pylori, you all know, it is an ulcer causing gastric pathogen and it is known to colonize the harsh acidic environments of the human stomach. So it survives in this particular acidic condition by producing urease, and this urease catalyzes hydrolysis of urea to yield ammonia, thus elevating the pH of its environment. So the answer is this chemical defense is hydrochloric acid. Answer is option B. Then which of the following pair of sugar is non-reducing in nature? So it is trehalose and sucrose, which are non-reducing. The trehalose is a non-reducing oligosaccharide in which the 2 D-glucose molecules are linked through anomeric position to one another. So it is through the anomeric carbon atom that the 2 D-glucose residues are linked in the case of trehalose, whereas in sucrose it is held together by glycosidic linkages between the C1 of alpha glucose and C2 of beta fructose. Since the reducing group of glucose and fructose are involved in glycosidic bond formation, sucrose is a non-reducing sugar. Then next one. The enzyme involved in the following reaction: amino acid plus ATP plus tRNA giving amino acyl tRNA plus AMP plus PPI. So it is amino acyl tRNA synthetase and is an enzyme that catalyzes amino acylation reaction by covalently linking an amino acid to its cognate tRNA in the first step of protein translation. So answer is option B, that is amino acyl tRNA synthetase. The next one, the resting membrane potential is determined by. So it is determined by K plus gradient. Answer is option A. The next one, when a robin eats an earthworm, the following happens. So earthworms, they are the major decomposers of dead and decaying organic matter. And the robin, it eats fruits, seeds, crushed peanuts, raisins, etc. but also feed on insects and worms. So here, earthworms eaten by robin, a plant eater, that is a earthworm which is a belongs to detrital food web, contributes energy to the plant eater robin. So the answer here is option A, that is detrital food webs can contribute energy to grazing food webs. Then which of the following is known as the sedimentary cycle because its reservoir is sedimentary rock? So it is phosphorus cycle. This is also a repeated question. Answer is option D. Then which of the following proteins bind to calcium during excitation, contraction, coupling? So it is the troponin. Answer is option D. Then next one. Both the substrates A and B must be present at the enzyme active site simultaneously is a characteristic feature of. So it is in the case of single displacement reactions. So single displacement reaction, it is of two types, that is ordered and random mechanism. Okay, there are two types, that is ordered and random mechanism. Ordered mechanism, the binding of the first substrate is apparently required for the enzyme to form the binding site for the second substrate. Okay, that is in the case of ordered mechanism in single displacement reaction. So both substrates A and B must be present at the enzyme active site simultaneously. It is a characteristic of simple displacement reactions. Then next one, number of nuclear pores depend on. So the number of nuclear pores, it depend on transcription activity of the cell. Answer is B. How many different linear tripeptides can be made from 3 different L-alpha neutral amino acid? Each amino acid only once in the chain. So these are the various combinations of amino acids that can be 3 different amino acids. Okay, leucine, isoleucine, alanine. L-alpha neutral amino acid, using each amino acid only once in the chain. So you can get how many? 1, 2, 3, 4, 5, 6. So 6 combinations are possible. So answer is option B. Then next one, the characteristic of organism involved in symbiotic relationships have developed through co-evolution. Involved in symbiotic relationship. Okay, it is symbiotic relationship, so it has developed through co-evolution. Then next one, M stripes, it is. PES is associated with one of the following. So M stripes, short for monitoring system for tiger intensive protection and ecological status. It is a software based monitoring system launched across Indian tiger reserves by Indian government's National Tiger Conservation Authority in 2010. And the system objective is to strengthen patrolling and surveillance of the endangered Bengal tiger. So it is associated with maintenance of tigers. Answer is option D. Then which of the following is not a method for enzyme immobilization? It is absorption. Absorption out of these is not a method of enzyme immobilization. Then next one, As part of the circadian clock, transfer of phosphate groups from adenosine triphosphate to PER is required. This is achieved by one of the following. So PER is period circadian clock gene is an important component of the circadian clock which modulates circadian rhythm. So the answer here is casein kinase 1. Answer is option A. Then in lake succession, the stages are observed in the following order. So the lower productivity is in oligotrophic state, then comes moderately productive mesotrophic state and highly productive euphoric state. So these are the stages in lake succession. So answer is option A, oligotrophic, mesotrophic and eutrophic. Then next one, the carrying capacity of a population is determined by its, so it is determined by its gross primary productivity. That is the gross primary productivity is the amount of carbon fixed during photosynthesis by all producers in the ecosystem. So gross primary production. Answer is option A. The next one. Chlorophyll B is distinguished from chlorophyll A by having a dash group in place of dash group. So chlorophyll B is distinguished from chlorophyll A by having a formyl group in place of methyl group. Answer is option C. Then next one. The proteins that are sorted to mitochondrial matrix are dash in nature. They are amphipathic in nature. The proteins that are sorted to mitochondrial matrix, they are amphipathic in nature. The next one, normal RBCs are, so biconcave in humans and in frogs. Frog it is biconvex, whereas humans it is biconcave. So that is not correct. Nucleated in camels and enucleated in humans. That is correct. Nucleated in camels and enucleated in humans. Then biconcave in camels and in frogs. Camel RBCs are oval in shape, not biconcave. Frog is biconvex. So the answer here, enucleated in humans and in camels. Answer is option B, that is nucleated in camels and enucleated in humans. Then next one. Which of the following pairs execute their function by binding with cytoplasmic receptors? So receptors for steroid hormones are usually found within the cytoplasm and are referred to as intracellular or nuclear receptors. So the answer is estrogen and progesterone. Noradrenaline is a neurotransmitter and a hormone. Growth hormone is a peptide hormone and GABA is a neurotransmitter. So the answer is estrogen and progesterone. They are steroid hormones. Answer is option D. Then artificial ripening of banana can be induced by ethylene. Answer is option C. Then diplospory and parthenogenesis lead to the development of. So diplospory is a condition where a diploid embryo sac is formed from megaspore mother cell without regular meiotic division. And parthenogenesis, reproduction from an ovum without fertilization. And parthenogenesis, it can operate either as a haploid or a diploid cell. So in haploid parthenogenesis, that is a rare form of pathogenesis and it occurs in few species of bees, nematodes and plants. And in such cases, the offspring develop from haploid egg to produce haploid adults. So answer is A and B. That is it can form haploid embryos as well as diploid embryos. Answer is option A and B. Then, which of the following is a true combination for Kala-Azar disease? So, Kala-Azar, it is caused by bites from female phlebotomine sandflies, the vector of the Leishmania's parasite. So, answer is option A, sandfly and Leishmania. Then next one, match the following and choose the correct combination. So sulfur is Methionine is having sulfur in its structure. Then zinc is oxine, magnesium is chlorophyll, and molybdenum is nitrogenous. So answer is option B. Then dash are the simplest transposable elements that contain inverted repeated sequences at each end and the gene encoding transposase. So they are the simplest transposable element and they are insertion elements. Answer is option B. Then the common feature of the following bacteriophages X174, OB, N4 and PI is they all infect E. coli. Answer is option D. Then protein synthesis in eukaryotes is inhibited by. So chloramphenicol and streptomycin, they inhibit protein synthesis in prokaryotes. Cephalosporin C, it disrupts the synthesis of the peptidoglycan layer forming the bacterial cell wall. So option D, that is cycloheximide, is a inhibit protein synthesis in eukaryotes. Then with reference to plant gynogenesis, with reference to plants, gynogenesis refers to. So gynogenesis is a process that leads to plant that exclusively originate from the female genetic background. So the answer here is development of haploid plants from the egg cell of unfertilized female gametophyte in vitro. Answer is option C. Then inclusive fitness is a measure of. So inclusive fitness is a method of measuring evolutionary success. It is the ability of an individual to transmit genes to the next generation, including genes shared with relatives. So the answer is the reproductive fitness of an organism and its relatives. That is option B. Then NMR is very important technique for determination of biopolymer structure. NMR experiments are not limited to the study of protons only. Which of the following represents a correct group of isotopes used to obtain resonance signal? Answer is D, that is H1, C13, N15, F19. Then next one. A linear DNA is 100% labeled at one end and has 3 restriction sites for EcoR1. If it is digested with EcoR1 so that the possible fragments are produced, how many of these fragments will be labeled and how many will be non-labeled? So a complete digestion, it means that all molecules that are present in the tube have been digested. But partial digestion means that all the molecules have not been digested. So there will be some completely digested, some incompletely digested molecules in the solution and they give rise to many different possibilities. So here we have got a linear DNA that is 100% labeled at one end. This labeling I have shown as a dot here. This is the label. Okay, and it has 3 restriction sites for EcoR1, that is E1, E2 and E3. So how many of these fragments will be labeled and how many will be unlabeled? So here you have E1. If it digests at E1, this will be digested, whereas E2 and E3 will not be digested. So E1 will be labeled, whereas E2 and E3 will not be labeled, or they will be unlabeled. And also one segment after E3 digestion. One another fragment also will be there. Then you have got E2 digestion. So it will digest at E2 but not at E1. So E2 region will be labeled, whereas E3 and this particular fragment it will be unlabeled. Then the next option is it will digest at E3 but not at E2 and E1. So you will get E3 which is labeled and this particular fragment which is unlabeled. Or else it cannot digest the DNA itself. Undigested DNA remains. So E1, E2, E3 all will be undigested and this will be the single band that will be labeled. So how many labeled you have? 1, 2, 3 and 4. Unlabeled 1, 2, 3, 4, 5 and 6. 6 unlabeled. So the answer here is option A, that is 4 labeled and 6 unlabeled. Then next one, cryopreservation is a process of. So cryopreservation, it is a process of ex situ in vitro conservation. Ex situ in vitro conservation. Answer is option A. So conservation of selected plants and animals in selected area outside their natural habitat is known as ex situ conservation. This is ex situ, this is cryopreservation. It is an ex situ in vitro conservation. Then next one, which of the following does not need an insert vector for transmission? So Rickettsia, Prow and C key, it requires the vector louse. Rickettsia, Rickettsy require the vector tick. Ehrlichia schaffensis is transmitted by ticks. And Coxiella burnetii, people get infected by breathing the dust that has been contaminated by infected animal feces, urine, milk and birth products that contain Coxiella burnetii. So Coxiella burnetii transmission doesn't need an insect vector. Answer is option D. The next one, hepatnaviruses, that is hepatitis B virus is an example, differs from other DNA viruses as their genome replication involves. It involves the action of the enzyme reverse transcriptase. Answer is option B. This was also a repeated question. I don't know for which exam they have asked this question, but it is repeated. Then in fermenter, the top portion left without the broth is called as the head space. Answer is option B. Then which of the following organisms get energy only by fermentative metabolism? So it is Lactobacillus bulgaricus. Answer is option A. Then next one, in the fertilized egg, cortical granule reaction required for slow block polyspermy is initiated by. It is initiated by Ca2+. Answer is option D. Then next one, when the velocity of enzyme activity is plotted against substrate concentration, which of the following is obtained? So it is a hyperbolic curve that is obtained. Velocity of enzyme plotted against substrate concentration, the usual Michaelis-Menten graph, it is a hyperbolic curve. The next one, anti-cancer drug cyclophosphamide is metabolized in the body by glutathione reaction. What is the type of this reaction? It is conjugation. Answer is option C. Then next one, in non-competitive inhibition. So in non-competitive inhibition, inhibitor will bind at a site other than the active site and may bind either to the enzyme or even to enzyme substrate complex. So answer is option B. Then mangrove swamps are found along. So they are found in tropical and subtropical coastlines. Answer is A. Then indicate the ionic species that predominates at pH 4, 8 and 11 for ammonia. So it is NH4 plus at pH 4, NH4 plus at pH 8 and NH3 at pH 11. Answer is option A. Then next one, which of the following transcription factor act as positioning factor for RNA polymerase II and binds to the promoter? It is transcription factor 2D. Answer is option C. Then TI plasmid is used in genetic engineering is obtained from. So it is obtained from Agrobacterium tumefaciens. Agrobacterium rhizogenus, it has RI plasmid. So answer is option D. Then next one, which enzyme of the following is used as a marker for lysosomal function? So it is acid phosphatase. Answer is option C. Then next one, the formation of Z ring in bacteria is important for. It is important for septum formation, that is during cell division, separation of the two daughter cells. That is option A. Then which of the following is not true of spermatogenesis? Answer is option D, that is differentiation of the gamete occurs while in diploid during the first meiotic prophase. So secondary spermatocyst divides into two equal haploid spermatids by meiosis II. So differentiation of the gametes occur while in diploid during first meiotic prophase is not the correct answer. Answer is option D. Then next one, a population of cells grown in adherent culture contain zero point 0.4 mg protein per 10 raised to 6 cells. Actin comprises 4.5% of the total protein. Given the molecular weight of actin is 42,000 Daltons and Avogadro number is 6.02 into 10 raised to 23, which of the following equals the mean number of actin molecules per cell? So you have got 0.4 mg in 10 raised to 6 cells and total protein is 0.4 and actin is 4.5% of the total protein. So 4.5 divided by 100 into 0.4 you will get 0.018 milligram in 10 raised to 6 cells. Now convert to per cell. This is 10 raised to 6 cells. So one cell will have 0.018 divided by 10 raised to 6. Now they have given 42,000 Dalton, that is the molecular weight of actin, and 1 Dalton is 1 gram per mole. So 42,000 Dalton will be 42,000 grams per mole. And 1 mole contains 42,000 grams of actin. And 1 mole will contain 4.2 into 10 raised to 7 milligrams of actin. And 4.2 into 10 raised to 7 milligram contains 6.023 into 10 raised to 23 molecules. So 0.018 divided by 10 raised to 6 milligram contains 2.58 into 10 raised to 8 molecules per cell. So the answer here is option C. Then next one. Which of the following is a natural dye obtained from plant? So it is hematoxylin. Answer is option B. Then next one. The group which are evolved from more than one ancestor are known as. So they are known as polyphyletic group. Answer is option C. Then which of the following is an example of a symmetrical animal? So hydra is, the sponges it is asymmetric, hydra is radially symmetric, frog is bilaterally symmetric and sea anemone is radially symmetric. So the answer which is asymmetric animal. This is not a symmetrical animal, it is A joined to S itself. So asymmetric animal it is sponges. So the answer is I think it must be sponges. I am not sure whether there is some mistake in the question also. Okay, whatever may be, if you take it as asymmetrical animal, it is sponges. Then next one. Which of the following chemical mutagens are incorporated into the genome by DNA polymerase during genome replication? So if you take alkylating agent, they are chemicals which are added to nucleotide bases by some chemicals. Example, EMS. Then deamination also chemicals like nitrous acid. Then intercalating agent, they produce mutations by sandwiching themselves between the adjacent bases. So they are acridine orange, ethylene bromide, etc. Then if you see for base analogues, that is it is a class of chemical mutagen that consists of base analogues. Base analogues means chemicals with structure similar to that of the any of the 4 standard bases of DNA. So DNA polymerase, it cannot distinguish between these analogues from the standard bases. So if base analogues are present during replication, they may get incorporated into the newly synthesized DNA molecule. So the answer here is option B, that is base analog. Next one, progesterone is synthesized in the hypothalamic neurons and stored in the posterior pituitary. So progesterone, it is an endogenous steroid hormone that is commonly produced by adrenal cortex as well as the gonads, which consist of the ovaries and the testes. And progesterone is also secreted by ovarian corpus luteum during the first 10 weeks of pregnancy, followed by placenta in the later stages of pregnancy. So A is not correct. Then progesterone plays a major role in preparing the uterus for implantation. That is correct. Then C is a protein hormone. It is not a protein hormone, it is a steroid hormone. So C is also not correct. Then is exclusively responsible for stimulation of FSH production and follicle growth. So it is actually gonadotropin releasing hormone stimulates pituitary gland to produce follicle stimulating hormone. So D is also not correct. Then next one. When somatic cells of ovule directly form embryos, the phenomenon is called. So we'll see what is adventive embryony. So adventive embryony, it is a type of apomixis in which development of embryos directly from the sporophytic tissues, that is nucellus and integument, take place. Then diplospory, it is a condition where the diploid embryo sac is formed from megaspore mother cell without regular meiotic division. Then apospory, it is the development of two end gametophytes without meiosis and spores from vegetative or non-reproductive cells of the sporophyte. And parthenogenesis, it is spontaneous development of an embryo from an unfertilized egg cell. So here they have asked, when somatic cells of the ovule directly form embryos, it is called as adventive embryony because it is a type of apomixis in which the development of the embryos develop directly from the sporophytic tissue, that is nucellus and integuments. So it is adventive embryony. Answer is option A. Then next one. Globin synthesis in the absence of heme in RBC is regulated by the phosphorylation of this factor. That is EIF2. Answer is option B. The next one. In anaerobic glycolysis, 2 moles of inorganic phosphate are used per mole of glucose consumed. Which of the following enzymes catalyzes the uptake of inorganic phosphate? So it is glyceraldehyde 3-phosphate dehydrogenase. Answer is option C. Then next one. When oxygen reacts with the Rubisco enzyme, the following products will be formed. So it is 3-phosphoglycerate and 2-phosphoglycolate. Answer is option A. It is oxygen reaction with Rubisco, it is photorespiration. Then next one. Which of the following is a functional excretory unit of cephalochordata? It is nephridia and solanocytes. Both A and C. Then Weismann proposed the theory of continuity of germplasm. Answer is option A. Then RAPD marker it is a dominant marker. Answer is option C. Then next, which of the following chromosomal aberration result in the formation of dicentric and acentric chromosomes? So it is paracentric inversion. Answer is option A. Then next one, in a hypothetical diploid animal species, there is a locus with 2 alleles A and B. If A and B are codominant, then how many different phenotypes are possible in the species? So it can have, this is just like our blood group. So it can have phenotypes AB, A and B. So answer is option C, that is 3 of them are possible. Then next one, if a woman having blood group A with AO genotype married a man having blood group with BB genotype, the expected proportion of heterozygotes among the children. So the woman is AO genotype and the man is BB. These are the gametes that are formed, that is A, O and B, and upon crossing you will get AB and BO. So they have asked for heterozygotes among the children. So these both are heterozygous. So it is 100%, that is the answer. So the answer is option D. Then next one. Type of migration where marine fishes migrate to fresh water for their reproduction is. So it is anadromous. Answer is option A. The propeller twist in B form of DNA is approximately. So what is propeller twist? It is that each base is planar. Okay, but when two bases pair, that is when they form base pair, they do not always line up perfectly flat with each other. This angle is called as propeller twist because the bases are twisted away from each other like an aeroplane propeller. So they have asked the propeller twist in B form DNA is approximately 36 degrees. Answer is option C. Then, the protein binding site in DNA can be identified by the following experiment. That is DNA footprinting. Answer is option A. Then Nobel laureate Barbara McClintock observed frequent breakage of maize chromosome 9 due to which of the following transposons? That is activator dissociator elements. Answer is option B. Then red-green color blindness is an example of dash genetic disorder. It is an example of X-linked recessive genetic disorder. Answer is option A. So that's it. Thank you for watching. Please do like, share and subscribe.